Now let's talk about heredity and genetics. Now, no two individuals are exactly alike. Even identical twins have differences. And one significant factor is heredity. So heredity is the passing of traits from the parents to the children. And traits that you inherit are things like eye and hair color, shape of your earlobes. And we need to understand environment can also influence inherited traits. For example, height and stature is an inherited trait, but poor nutrition may stun a child's growth. So chromosomes and genes. Most cells of your body contain a nucleus. This is the cell's control center. Inside each nucleus is a set of chromosomes, which are thread-like structures found within the nucleus of a cell that carry the code for inherited traits. Most cells in the body contain 46 chromosomes arranged in 23 pairs. Now, sections of chromosomes called genes carry codes for specific traits. Genes are the basic unit of heredity. Like chromosomes, genes occur in pairs. One gene from each pair is inherited from each parent. You have thousands of genes in every cell of your body. So let's talk about DNA. DNA is a chemical unit that makes up chromosomes. All living things are made of DNA. Chemical compounds called bases make up the structure of DNA. The arrangement of the bases along each DNA molecule differ. Because several thousand pairs of bases are in each gene, countless numbers of arrangements are possible. The order of the bases is called a genetic code. Cells use a genetic code to make proteins. Proteins help to build and maintain body tissues. Different kinds of proteins result in various individual traits. All the characteristics that you have, the color of your eyes, the amount of curl in your hair, are determined by your genetic code. Unless you have an identical twin, your DNA is different from that of any other person. So now let's talk about genetics and fetal development. Okay, each living organism has a certain number of chromosomes. Although most humans contain 46 or 23 pairs, like we said previously, sperm and egg cells have only half that amount, or 23 chromosomes. When a sperm and egg unite, the resulting zygote will have 46 chromosomes, or 23 from each parent. These chromosomes carry the hereditary traits of the parents. As we talked about earlier, a zygote divides many times, producing trillions of cells that make up the human body. Between each cell division, each chromosome in the cell nucleus duplicates itself, producing two sets of 46 chromosomes. As a cell divides, the two sets of chromosomes separate. Each new cell will contain one set of 46 chromosomes that are identical to those in the first cell of the zygote. So let's talk about dominant and recessive genes. So at least one pair of genes is responsible for each human trait. Some genes are dominant and others are recessive. Now the traits that are dominant, or the genes that are dominant, generally appear in offspring whenever they are present. The traits of a recessive gene usually appear only when dominant genes are not present. For example, suppose an individual receives two genes for eye color, one for brown eyes and one for blue eyes. The resulting individual will have brown eyes because brown eyes is a dominant gene, where blue eyes are a recessive gene. An individual with blue eyes must have two recessive genes for blue eye color. Now the situation is more complex than in the example above, because traits that express a quantity or an extent, such as height, weight, or color, usually depend on many gene pairs, not just one. Now let's talk about genes and gender. In humans, one pair of chromosomes determines the gender of an individual. If you are female, these two chromosomes look exactly alike and are called X chromosomes. If you are male, the two chromosomes differ. One is shorter than the other. The shorter chromosome is the Y chromosome. The longer one is the X. Remember that sperm and eggs contain only half the chromosomes of other cells, or one sex chromosome, not two. Sperm contain an X or a Y chromosome. Eggs have and X chromosome. So the gender of a child is determined by which type of sperm, X or Y, unites with the egg. So it's the sperm that determines the gender of an offspring. Now let's talk about genetic disorders. So some of the genes that an individual inherits contain a mutation or an abnormality in the base sequence of the genetic code. Often the mutation has little or no effect on the individual, but sometimes the mutation can result in defects or other health problems called a genetic disorder. So genetic disorders are disorders caused by partially or completely by a defect in genes. Some genetic disorders, such as those that cause birth defects, are apparent right away. One example of such a defect is a cleft palate. However, other genetic disorders do not show up until later. So some other genetic disorders. We have sickle cell anemia. This is when red blood cells have a sickle shape or a curved shape and clump together, may result in severe joint and abdominal pain, weakness, kidney disease, and restricted blood flow. We have Tay-Sachs disease, which is destruction of the nervous system, can lead to blindness, paralysis, or death during early childhood. We have cystic fibrosis, so when mucus clogs many organs, including lungs, liver, pancreas. You have nutritional problems, serious respiration infections, and congestion. Down syndrome. Now, there's varying degrees of mental retardation, short stature, round face with upper eyelids that cover inner corners of the eyes. Then we have hemophilia, which is the failure of the blood to be able to clot. So, how do we identify if someone's potentially going to have a genetic disorder? Because although most genetic disorders cannot be cured, in some cases they can be treated, especially if they're diagnosed early, often before birth. So two common technologies used to test for genetic disorders are an amniocentesis, which is a procedure in which a syringe is inserted through the pregnant female's abdominal wall into the amniotic fluid surrounding the developing fetus. Doctors can then examine the chromosomes in fetal cells taken from the amniotic fluid for genetic and check for genetic abnormalities or to determine the gender and age of the fetus. Amniocentesis is usually performed 16 to 20 weeks after fertilization. And you also have chronic villi sampling, or CVS, which is a procedure in which a small piece of membrane is removed from the chorion, a layer of tissue that develops into the placenta. The tissue can be examined for genetic disorders or to determine fetal age and gender. The procedure is done around 8 weeks of fetal development. Tests for genetic disorders may also be done after a child is born. For example, many states require the testing of all newborns for PKU, which is diagnosed soon after birth. A baby's diet can be altered to stop possible mental retardation caused by this genetic disorder. Then we have genetic counseling. So research for diagnosing, preventing, and treating genetically related disorders has resulted in a wide variety of programs. Genetic counselors can advise families about the probability of having a child with genetic related disease. They also can guide families of children with genetic disorders about possible treatment options. And now we also have genetics and research. So we can use genetic research to help cure diseases. So we have gene therapy. Many disorders result with one individual's lack a functioning gene. And without the functioning gene, certain substances that the body needs cannot be produced. So gene therapy is the process of inserting normal genes into human cells to correct genetic disorders. When the defective gene is replaced with a normal one, the cells with the new gene begin to make the missing substance. Most often viruses are the carriers used to insert the new gene into a person's cell. The practice of placing fragments of DNA from one organism into another is called genetic engineering. And it is considered highly experimental. Genetic diseases for which scientists are researching gene therapies include cystic fibrosis and various cancers. Then we have genetically engineered drugs as well. So genes are used to treat diseases, aren't always inserted directly into the human beings. Instead, they are placed into other organisms, causing them to produce substances that can be used to treat human diseases or disorders. And so generically produced medicines include treatments for burns. Genetically produced medicines include treatments for burns, ulcers, growth defects, ovarian and breast cancers. So genetic engineering also is used to produce some vaccines that prevent diseases. So now let's talk about infancy and childhood. So there's dramatic physical and mental changes that take place as an infant grows through childhood. There's a lot of different theories of how children should develop, but one of the most widely accepted theories of development is that of psychologist Erik Erikson. So let's talk about childhood development. So each stage is characterized by developmental tasks, events that need to happen in order for a person to continue growing towards becoming a healthy, mature adult. Success in each stage is dependent on an individual's experiences during that stage. Partial development at one stage can be overcome by developmental success in the following stage. Now we're going to talk about the 4 stages of infancy and childhood right now. So infancy, which is considered birth to 1 year. So at this stage, humans are just learning to develop trust, to trust other people because they're reliant on other people. So an infant is completely dependent on others to meet his or her needs, and they must be able to trust others to provide for their needs. Infancy is the period too where the fastest growth in a person's life. During this time, a child's his or her weight will triple, and his or her weight might increase by 50%. Infancy is a time for learning how to eat solid food, how to sit, crawl, walk. Then we have stage 2, which is early childhood. This is 1 to 3 years. At this stage, children should be developing the ability to do tasks by themselves. And so children in early childhood begin to feel proud of their accomplishments, and they become eager to learn more. During this time, children develop many new skills. They learn to talk, climb, push, pull. They increase their vocabulary and begin talking in sentences. If parents accept the child's needs to do whatever he or she is capable of, the child can develop a sense of autonomy, which is a confidence that a person can control his or her own body, impulses, and environment. If a parent is overprotective or overly critical of a child's behaviors, the child may develop doubts about his or her abilities, and it can stunt their development. Then we have stage 3, which is middle childhood, which is 3 to 6 years. And now during this stage, children learn to initiate play activities rather than merely following the lead of others. Children of this stage also display their intelligence by asking many questions. They must learn to recognize emotions and practice expressing them in appropriate ways. If parents show approval of these new abilities and encourage questions, children learn creativity, initiative, and the ability to start something on their own. Children of parents who are impatient with the child may develop a sense of guilt about self-initiated activities, resulting in low self-esteem. And so the big part of this is they have to develop responsibility, take initiative, and create their own play. Then we have stage 4, which is late childhood, which is 7 to 12 years. Now during late childhood, school becomes an important part of a child's life. Children develop skills like reading, writing, math. They also learn how to get along with peers, learn appropriate roles in society, and develop a conscience. If their efforts are rewarded and appreciated, their pride in their work increases. Children who are scolded for making a mess, getting in the way, or not following directions may develop feelings of self-doubt. And so in that late childhood, we were trying to develop an interest in performing activities, and the child learns transitions from home to school, learns how to make things, use tools, and acquires new skills. So health screenings in children are important. Vision and hearing impairments can affect a child's development as much as social factors do. Immunizations and health screenings can prevent many problems. According to the CDC, nearly 1 in every 1,000 children in the United States has low vision or is legally blind. Schools often provide regular vision screenings for students. Now, in the United States, 2 to every 3 of every 1,000 infants are born with hearing impairment, severe enough to affect language development. Some state laws require newborns to be screened at birth for hearing loss, and schools often provide periodic screenings. Now, we also screen for scoliosis, which is something we talked about previously this semester. Now, scoliosis, to remind you, is an abnormal lateral or side-to-side curvature of the spine. It may begin in childhood and go unnoticed until the teens. Its exact cause is unknown, though it is more common in girls. And many public schools make it a policy to check for scoliosis in middle school. And so that concludes Chapter 19. Use this to get through the first half of Chapter 19 and 20 worksheet, and check back on Wednesday for the Chapter 20 presentation.